Welcome to this edition of Research Connection. Joining us today is Kathryn Polkoff, or maybe I should call it Quarantine Connection, Kathryn.
That's where we're at right now.
So what are you sitting in front of?
Right now, I am sitting in front of a hair follicle, as you might imagine. I feel like Mrs. Frizzle, honestly. We're deep in the skin. And so this is something I'm going to talk to you a bit about today.
Yes, so tell us a little bit about yourself.
I am a PhD student working with Jorge Piedrahita, and I am studying hair follicles in the skin. I'm from Chicago, Illinois, and I went to University of Illinois for undergrad, and I did a master's in animal science there before deciding to come here in the department or the program of comparative biomedical sciences for my PhD.
Neat. And you recently received an F award from the NIH.
Yeah, that's correct. I got the F-31, which is very exciting for me, and I think for the college. It was quite an experience to kind of apply and put together a team of advisors that were going to be really helpful to the whole experience.
So what's the research on?
So the research is on the hair follicle, I guess. So the idea is that in the skin, in the epidermis, we know that there are several different populations of stem cells, and one of the particular types of stem cells is found only in the hair follicle. And so in this picture, they're labeled with the green marker. So they're found in your hair follicles, and they stay in your hair follicles, and they give rise to your hair, and they're responsible for regulating hair growth and recycling. But they never really leave the hair follicle unless there is an injury in the vicinity. And then those cells are found to migrate from the hair follicle and then go into the wound and cover and make a new epidermis. But no one actually really understands what causes them to do it, when do they go, when do they migrate, and how we can harness those as a therapy. So the grant proposal in the F-31 is in efforts to understand what factors draw those cells from the niche into the wound, and then how can we kind of harness these therapeutically, and what compounds would drive this reaction in vivo. So we use a transgenic pig as a model, and so the idea is that the cells at the base of the hair follicle that are in green here, this is actually GFP in a hair follicle of a pig. So it's natural GFP. We didn't stain it except for the red, which is the Ki67. But we use this pig so we can trace the cells in real time, and then we can extract the cells and sort the cells and see what kind of factors they're secreting or messages they're making based on RNA.
That's fascinating. I have a new appreciation for hair now.
Right, might just be a new skin graft.
And I guess an interesting question is, has the quarantine impacted this research at all? Are you delaying it?
Yeah, it's because a lot of it is reliant on pigs and our transgenic pigs, and we can't really be in the lab to process their tissue or to analyze what we can do. It's kind of put a halt on the actual progress of the research, but it's allowed us to kind of reflect on what we have so far and write the papers and kind of think of new proposals for grants. Our lab has done something really cool where we just kind of have lab meetings once or twice a week, and we just brainstorm for 2 hours, and everyone read the paper. It's like a journal club, but we've all read a little bit of background. So that's been really fun in this quarantine, just like a big think tank.
Yeah, that's good to hear. And life goes on, right? You said more pigs have been born since, so.
Yeah, we had 2 litters born in the quarantine, so I guess we could come up with cool COVID names for them. I don't know.
Well, neat. Well, thank you for sharing. Best of luck. Take care.
Thank
Stay
you.
safe. And
Too.
hope to see you soon.
I hope so.